Today we're going to be talking about pre-translational modifications that are undergone by the eukaryotic pre-mRNA. Playing the part of pre-mRNA is Revelstoke the dog. Let's get started. One quick note about our star Revelstoke the dog before we begin. Revelstoke is a willing participant in this project and receives cheese throughout. However, because Revelstoke is 25% Chihuahua, he often appears to be having an existential crisis even while actively receiving treats. I just want to reassure you that no sweet Revelstokes were abused during the making of this educational video. Thank you for watching. Let's discuss context. We are going to be looking at a process which is occurring inside of a eukaryotic cell. Inside of the nucleus after transcription but before translation. There are a number of important differences between the mRNA handling of a eukaryotic versus a prokaryotic cell, and this video is just going to be discussing the 3 modifications required for an mRNA before that mRNA can exit the nucleus and be involved in translation. Now back to Revelstoke the dog showing us how this all works. Modification number 1 is to remove this orange interron vest through mRNA splicing. This is accomplished through a series of events in which the exons are connected and the interron is removed, forming a lariat structure. Modification number 2 is the addition of a poly-A tail. This is a long chain of the nucleotide adenine. Each bow stands for about 10 to 15 adenines, making the tail 150 to 200 adenine groups long. This poly-A tail modification prevents degradation once the pre-mRNA leaves the nucleus, allowing more time for translation to occur. It also increases the stability. The third and last modification is the addition of a 5' guanine cap. This 5' guanine cap marks the site of initiation of translation. Once the eukaryotic pre-mRNA has received all 3 of these modifications, it will be allowed to exit the nucleus and to begin translation. Big thanks to Revelstoke the dog for helping us film this video today.